Hi everyone, my name is Natalie and I have the pleasure of giving you guys a presentation on the overview of glycolysis and gluconeogenesis with a focus on aldolase B. So the objectives for this presentation is basically to be able to describe the overall pathways of glycolysis and gluconeogenesis and explain how they work in opposite directions. Explain how the role of aldolase in both pathways. Differentiate between the three aldolase isoforms. Discuss why aldolase B is essential in the liver for both gluconeogenesis and fructose metabolism. Describe how aldolase B expression is regulated by transcription factors including HNF1, HNF3, CEBP, and DBP. So first things first, when it comes down to glycolysis and gluconeogenesis, our daily movements require ATP, which is produced by the oxidative catabolism of D-glucose. So glycolysis begins with D-glucose and ends with 2 molecules of pyruvate and a net of 2 ATP, and gluconeogenesis begins with pyruvate and ends with D-glucose. So we're going to go over the steps of glycolysis. We can break them down into 2 different phases. The first phase, we can call it the preparatory phase or the investment phase, and the second half of glycolysis, we can call it the investment phase. And throughout this presentation, you will understand why there's 2 different phases and what do they mean exactly. So glycolysis is a degradation of glucose, hence the name glycolysis, right? So it begins with glucose, and that glucose is phosphorylated by the enzyme hexokinase. Hexokinase grabs that phosphate group from ATP and transfer it over to the 6th carbon position on glucose. And now we have glucose 6-phosphate. And this is the first ATP that is invested in this pathway. Then we have isomerization that takes place by the enzyme phosphoglucose isomerase. It converts glucose 6-phosphate into fructose 6-phosphate. Then another phosphorylation takes place with the enzyme phosphofructose kinase. Again, it grabs that phosphate group from ATP and transfers it over to fructose 6-phosphate. And now we have fructose 1,6-bisphosphate. Now, an important thing to understand about this step is that this step is irreversible, meaning it cannot go back into the reverse pathway. You'll know that when we cover gluconeogenesis. But also, PFK cannot continue its job. It's what it's meant to do if there's high levels of ATP and citrate in the cell. So PFK can only be signaled to phosphorylate fructose 6-phosphate if the ATP levels and citrate levels remain really low in the cell. So basically high levels of ADP. So if phosphofructose kinase gets the signal to go ahead and phosphorylate fructose 6-phosphate, it then goes on and of course make fructose 1,6-bisphosphate, which is cleaved by the special enzyme aldolase. And this cleavage takes place, and we have 2 triose phosphates. We have dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Now, this is an interesting step because when it comes down to these 2 triose phosphates, when it comes down to equilibrium, dihydroxyacetone phosphate is favorable. It's more favorable, about 95%. But for some reason, only glyceraldehyde 3-phosphate is pushed in the forward direction to basically continue on with glycolysis. And why does this take place? So it's interesting to know that the enzyme glyceraldehyde 3-phosphate dehydrogenase basically consumes glyceraldehyde 3-phosphate so quickly and therefore causes the enzyme triose phosphate isomerase to push dihydroxyacetone phosphate to continue to be converted into glyceraldehyde 3-phosphate. And this happens very quickly. So this is why glyceraldehyde 3-phosphate is pushed towards glycolysis and dihydroxyacetone isn't. It's not saying that it's not being used, but its sole purpose in this reaction, in this pathway, is to be converted through isomerization. So once the enzyme GAPDH basically oxidizes the glyceraldehyde 3-phosphate, it gets converted into 1,3-bisphosphoglycerate, and we have this inorganic phosphate group that is attached to it now, and this produces NADH. And then we have the enzyme PGK that goes ahead and dephosphorylates the 1,3-bisphosphoglycerate, and now we have 3-phosphoglycerate and also a net of 2 ATP. Not a net of 2 ATP, I'm sorry, I apologize, but a 2 ATP is produced with this reaction. And I didn't mention earlier, but this reaction is known as the payoff phase, and it's because previously with the first half of glycolysis, it was investing ATP. You saw a total of 2 ATP invested in the first 5 steps, and now you'll see ATP being produced in the remaining steps of this pathway. Then we have phosphoglycerate mutase, which basically rearranges the phosphate. It was first on the 3rd carbon, and now it's on the 2nd carbon. Then enolase goes ahead and do a dehydration action, a dehydration mechanism, removes the hydration from 2-phosphoglycerate, and now we have phosphoenolpyruvate. And then dephosphorylation takes place with the enzyme pyruvate kinase, and there are no more phosphate groups on the molecule, and now we have singular pyruvate, but also 2 molecules of pyruvate and also 2 ATP that's being produced. So in total, we have 2 ATP invested in the first half of glycolysis, 4 ATP being produced in the payoff phase, and in total, this is a net of 2 ATP. So when it comes down to gluconeogenesis, it occurs when glucose level falls. It basically synthesizes glucose from non-carbohydrate precursors such as lactate, pyruvate, and other amino acids. And what triggers gluconeogenesis is our high levels of acetyl-CoA. So our muscles, basically other cells or the tissues, can basically function off the energy that's being given off when fatty acids are being broken down. However, our brain and our blood cells cannot rely on that fatty acid energy. It needs glucose. So because of this, our cells have I wouldn't say came up with a way, but there's a mechanism for glucose to still be produced when glucose is not being intaken. So gluconeogenesis just means the formation of new sugar. And before this can happen, I did mention that gluconeogenesis, it needs to start with high levels of acetyl-CoA. That's because it's a signal that fatty acids are being broken down, energy is coming from it, and also is signifying that there's a fasting going on. So what happens is that the enzyme pyruvate carboxylase, it goes ahead and converts pyruvate to oxaloacetate after it receives the signal that there's high levels of acetyl-CoA within the cell. Now, once pyruvate is converted into oxaloacetate, something interesting happens is because it can't be transported out of the mitochondria into the cytosol. Now, it gets reduced into maltate. Now, when it gets reduced into maltate is then transported out of the mitochondria, because maltate has transporter proteins, unlike oxaloacetate, to transport it out of the cytosol. Once that happens, maltate is in the cytosol. It is then reoxidized back into oxaloacetate, and then the enzyme PEPCK, phosphoenolpyruvate carboxykinase, go ahead and basically phosphorylates and convert it into phosphoenolpyruvate using 2 ATP. Now with this is basically one version of this mechanism. Now, if it starts with a lactate and not pyruvate, the reducing of oxaloacetate doesn't take place. Why? Because there are high levels of NADH present in the mitochondria. So what happens if lactate is the precursor instead, the 3-carbon molecule that's being used, lactate would then be converted into pyruvate. Pyruvate would then be converted into oxaloacetate, and right there, oxaloacetate would be then converted into phosphoenolpyruvate. It would not need to be reduced into maltate and then reoxidized. It would just go, basically, it would just happen with ease. There's no need for transporting it out. But since we're talking about pyruvate is the start of this pathway, so that's we're going to keep it that way. And then we have the enzyme enolase that is going to hydrate the phosphoenolpyruvate, and we're going to give us 2-phosphoglycerate. And then the enzyme phosphoglycerate mutase is just going to rearrange those phosphate groups, and then we have 3-phosphoglycerate. And then the enzyme phosphoglycerate kinase is going to basically phosphorylate that phosphoglycerate, and now we have 1,3-bisphosphoglycerate. Now again, GAPDH in glycolysis, it oxidizes those glyceraldehyde 3-phosphate, but now in gluconeogenesis is going to reduce 1,3-bisphosphoglycerate to give us those isomers, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. And then our special enzyme, again, aldolase, is going to basically combine those triose phosphates, those 2 triose phosphates, and give us fructose 1,6-bisphosphate. And then fructose 1,6-bisphosphate is going to basically dephosphorylate fructose 1,6-bisphosphate and give us fructose 6-phosphate. And then the enzyme phosphoglucose isomerase is going to convert the fructose 6-phosphate into glucose 6-phosphate. And lastly, we have glucose 6-phosphatase, that's going to give us our free glucose. That's going to enter the bloodstream. Now, I do want to mention, which I almost forgot, is that there are some bypass phases here in gluconeogenesis. Reason being is because in glycolysis, there are some irreversible steps that takes place, and it will be physically impossible to basically go in the backwards or the reverse pathway to perform those same exact steps. So because that's physically impossible, gluconeogenesis uses different enzymes to kind of perform the same thing, but not using those specific enzymes. So one of those steps that are bypassed are basically pyruvate to phosphoenolpyruvate. Another is fructose 1,6-bisphosphate to fructose 6-phosphate. That's a second bypass step. And the last bypass step is glucose 6-phosphate to glucose with the enzyme. So each of these enzymes that are being used with these bypass steps, the 3 bypass steps that are mentioned, are not used in glycolysis. Now, when it comes down to the regulation of glycolysis and gluconeogenesis, just need to understand that the liver is the key regulator for maintaining the blood glucose levels. When it comes down to glycolysis, it keeps our blood glucose levels low by using it. Again, glycolysis is the breaking down of glucose. And when it comes to gluconeogenesis, I wouldn't say it increases the blood sugar level, but it keeps it at a higher level versus when someone is fasting or when the cells are fasting. Excuse me. So a recap on aldolase, because we are going to speak more about aldolase because it is a special enzyme, even though it's not part of the regulatory enzymes when it comes down to the pathways, but it is very important. Just to refresh the memory, aldolase is basically in glycolysis, it cleaves the fructose 1,6-bisphosphate into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. But in gluconeogenesis, it synthesizes it basically, or I want to say put them together and forms the fructose 1,6-bisphosphate. So again, aldolase is a very important enzyme in the glycolytic pathway and even in gluconeogenesis, and it also comes in different forms, different isoforms called aldolase A, aldolase B, and aldolase C. And each isoform is found in different tissues. So aldolase A is expressed largely in muscle tissue and red blood cells. Aldolase B is expressed more in the liver, and you find some of it in the kidney, but not as much as in the liver. And aldolase C is expressed mainly in the brain. So the main differences between these isoforms, they do differ in about in their amino acid sequence, about 20% different from each other. However, these differences do make a big change. It does make a big difference. That can go from the hydrophobic areas, the regions that are hydrophobic, the regions that are not hydrophobic. also the Vmax, the catalytic activity that it can perform. And especially with aldolase B, we're going to but I'm going to explain to you and later on in the presentation that aldolase B is specifically the only enzyme that's used to catalyze a specific molecule. So those are the differences between the different isoforms of aldolase, which is pretty interesting. And lastly, we have like aldolase A, which has the highest glycolytic activity and binds fructose 1,6-bisphosphate more tightly, which makes it efficient for breaking down glucose in the muscle tissue. Now, an interesting fact that I want to mention about aldolase is that both aldolase A and aldolase B mRNA are present before birth, but after birth and throughout adulthood, only the liver is primarily expressing aldolase B, which becomes a dominant form. And again, aldolase B is extremely important because it needs to basically cleave the fructose 1,6-bisphosphate into the triose phosphates, or it needs to form the fructose 1,6-bisphosphate from the triose phosphates. And it's important because how can we get, especially when it comes down to fasting and gluconeogenesis, how can we get that glucose to be produced and sent off into the bloodstream if we do not have a 6-carbon to start with? And that fructose 1,6-bisphosphate is what help us get that glucose at the end. Now, when it comes to regulation, we do have some transcription factors that do play a role in basically ensuring that there's enough aldolase B expression and during specific times, like fasting times, or even throughout sleeping in. We will cover that throughout the presentation. We have HNF1 and we have HNF3. So HNF1 is a strong activator of aldolase B promoter regions. It binds to the promoter regions and ensures high expression of aldolase B, and it makes sure that it's specific to the liver. And it also, you can imagine if there's a form of, let's say, a mutation or something's wrong with this HNF1 transcription factor, then you can say that it will affect how much aldolase B is expressed. Now, because of that, there can be a cascade of events that can be harmful to the cell. If there's not enough aldolase B expression, then we can conclude that there won't be a way to break down fructose 1-phosphate. And now what does this have to do with anything? Because fructose 1-phosphate is not mentioned in glycolysis nor in gluconeogenesis, but it's important in the liver. In the liver, we find fructose 1-phosphate. However, aldolase B is the only enzyme that can break it down. And you can imagine if there aren't any expression, if there aren't any HNF1 basically binding to the promoter region and activating that expression of aldolase B, then we won't be able to break down fructose 1-phosphate. And that buildup in the liver can cause damage to the liver cells. It can also cause ATP depletion or lack of ATP, and it can also cause other damages like hypoglycemia. So the next transcription factor, we have HNF3, which acts as a stabilizer. So you can view HNF1 as an activator and HNF3 as a stabilizer. It basically maintains aldolase B expression during fasting when the liver shifts to glucose production, ensuring that the enzyme is available during prolonged exercise or low glucose condition. So again, the difference between the two transcription factors, we have one, HNF1, that's an activator turning aldolase B expression on, and we have one that's a stabilizer that's making sure that the expression of aldolase B is being maintained when the liver needs glucose the most. So the last two transcription factors that I want to cover that basically have a layer of control over aldolase B expression are CCAT enhancer binding protein and Gbox binding protein. So when it comes down to CCAT enhancer binding protein, it becomes more active during metabolic stress or fasting, especially when glucogen and cortisol levels rise. So its role is to make sure the liver has enough aldolase B available to produce glucose when the body needs it the most. Not when it has sufficient amount of glucose, but only when it needs it the most. So essentially, it coordinates aldolase B with other gluconeogenic enzymes so glucose production can meet its energy demand. And those enzymes are phosphoenolpyruvate carboxykinase, fructose 1,6-bisphosphatase, and glucose 6-phosphatase. And if you remember, these are the specific enzymes that are used in gluconeogenesis that are using gluconeogenesis as the bypass enzymes. Because in glycolysis, they don't have these enzymes. So you can imagine if there's a issue with the CCAT enhancer binding protein, if there's like a mutation, not only would it not be able to express aldolase B during fasting times, routine fasting, but also it won't help express these enzymes that are used in gluconeogenesis, which will be a big issue because without these enzymes, gluconeogenesis cannot proceed. And lastly, we have the D-box binding protein, which acts like a switch, like an on and off switch, a circadian transcription factor. So what it does, it binds to the D-box region of aldolase B, promoter, and adjust expression based on the time of day. This means that aldolase levels are higher during fasting periods like night and lower when food is being consumed. So you ever wonder that, like, when you go to sleep, why don't you feel hungry? Well, some people do feel hungry, but your body is not supposed to keep you up hungry throughout the night because it understands that around this time period, let's say you go to sleep at 11 o'clock every day at night. So from 11 o'clock until, let's say, you wake up at 8 a.m., your body is going to undergo gluconeogenesis to produce more glucose because it understands that glucose is not being consumed at this point. It understands that. And the D-box binding protein transcription factor is pretty much plays a big role in this. And when you wake up at 8 a.m., your body expects you to basically eat something, and therefore it undergoes glycolysis. And you can do this experiment with yourself to see if it's actually true. I do this with myself all the time. Let's say if I'm doing a fast, if I'm fasting from 9 to 12 or something like that, usually I would eat breakfast like around 9 o'clock. But when I do it abruptly, when I go into a fast abruptly, my body goes haywire. But after a week or so, my body gets used to it. And I can definitely say I can basically give it to this transcription factor as a help for that. So in short, DPP ensures that aldolase B is expressed in sync with our metabolic needs, while the CCAT enhancer binding protein ensures that aldolase B is basically ramped up during fasting or stress. And it seems like they're both are doing the same thing, but one of them is specifically used when we fast, and the other one is used when it's a cycle of fasting, so sleeping. And I'd say if your cells already know what time of day you're going to be working out, and those type of instances. Now, just to summarize this entire presentation, we spoke about the degradation of glucose, which is glycolysis. We spoke about how there is two different phases of glycolysis. We have the investment or the preparatory phase, and we have the payoff phase, which means that 2 ATP are invested and 4 ATP are paid off, and a net 2 ATP are produced throughout this whole glycolysis pathway. And glycolysis also have these irreversible steps that cannot be used in gluconeogenesis. Gluconeogenesis is not specifically just glycolysis in the reverse. They do have some steps that are bypassed because in glycolysis, it would be very difficult, physiologically impossible to go in the reverse direction of these pathways because of the large negative delta G. And because of that, gluconeogenesis uses different enzymes to basically go around those bypass steps to continue in the reverse pathway. We also basically covered how the liver uses gluconeogenesis when it's shifting into fasting. We focused on aldolase with the emphasis on aldolase B and how it's very important. Aldolase B is the only enzyme that can break down fructose 1-phosphate. Without it, there will be a buildup of fructose 1-phosphate in the cells, in the liver cells. We spoke about the different transcription factors that play a key role in aldolase B expression: HNF1, HNF3, the CCAT enhancer binding protein, and DPP. So hopefully you're able to answer these questions with this presentation. This serves as a knowledge check. So the questions are: name the 3 irreversible glycolysis steps that are bypassing gluconeogenesis and explain why. How many isoforms of aldolase are present? Aldolase B is found abundantly in what type of cells? What role does the transcription factor DBP play in regulating aldolase B expression? and how does this relate to the body's daily metabolic cycles? So again, you should be able to answer these questions based on this presentation. And if not, then I guess you can do some more research, but the information in this presentation should be enough to answer them. Thank you guys so much for tuning into the presentation, for watching. Thank you.